Receptors of insects. All insects are sense organs that allow them to see, smell, taste, hear, and touch their environment. Since these are the same five senses we humans experience, it is tempting that insects see what we see, hear what we hear, smell what we smell, etc. Experimental evidence has shown that an insect's sensory capabilities are very different, both quantitatively and quantitatively, from those of humans and other vertebrates. All sense organs receptors act as transducer, converting light energy, chemical energy to mechanical energy from the environment into electrical energy of nerve impulses in sensory neurons. Signals generated by insect sensory receptors travel to the brain or ventral nerve cord where they stimulate appropriate behavior responses. Finding resources, food, mate, etc., avoiding danger or reacting to changes in the environment. Finding resources, food, mate, etc., avoiding danger or reacting to changes in the environment. Sense organs, chemical senses include the use of chemoreceptor related to taste and smell affecting mating, habitat selection, feeding, and parasitic host relationships. Taste is usually located on the mouthparts of the insect, but in some insects, such as bees, wasps, and ants, taste organs can also be found on the antennae. Taste organs can also be found on the tarsi of moths, butterflies, and flies. Olfactory sensilla enable insects to smell and are usually found in the antennae. Chemoreceptor sensitivity related to smell in some substances is very high, and some insects can detect particular odors that are at low concentrations miles from their original source. Mechanical senses provide the insect with information that may direct orientation, general movement, flight from enemies, reproduction, and feeding, and are elicited from the sense organs that are sensitive to mechanical stimuli such as pressure, touch, and vibration. Hairs, setae on the cuticle are responsible for this as they are sensitive to vibrational, touch, and sound. Hearing structures or tympanal organs are located on different body parts such as wings, abdomen, legs, and antennae. They can respond to various frequencies ranging from 100 to 240 kHz depending on insect species. Many of the joints of the insects have tactile setae that register movement. Hair beds and groups of small hair-like sensilla determine proprioception or information about the position of a limb and are found on the cuticle at the joints of segments and legs. Pressure on the body wall or strain gauges are detected by the campaniform sensilla and internal stretch receptors sense muscle distension and digestive system stretching. The compound eye and ocelli supply insect vision. The compound eye consists of individual light receptive units called ommatidia. Some ants may have only one or two. However, dragonflies may have over 10,000. The more ommatidia, the greater the visual acuity. These units have a clear lens system and light-sensitive retina cells. By day, the image flying insects receives is made up of a mosaic of specks of differing light intensity from all the different ommatidia. At night or dusk, visual acuity is sacrificed for light sensitivity. The ocelli are unable to form focused images but are sensitive mainly to differentiate in light intensity. Color vision occurs in all orders of insects. Generally, insects see better at the blue end of the spectrum than at the red end. In some orders, sensitivity ranges can include ultraviolet. A number of insects have temperature and humidity sensors, and insects being cool small more quickly than insects being small cool more quickly than larger animals. Insects are generally considered cold-blooded or ectothermic, but their body temperature rising and falling with the environment. However, flying insects raise their body temperature through the action of light above environmental temperatures. The body temperature of butterflies and grasshoppers in flight may be 5 degrees Celsius or 10 degrees Celsius above environmental temperature. However, moths and bumblebees, insulated by scales and hair during flight, may raise flight muscle temperature 20 to 30 degrees Celsius above the environment temperature. Most flying insects have to maintain their flight muscles above a certain temperature to gain power enough to fly. Shivering or vibration with wing muscle allow larger insects to activate, increase the temperature of their flight muscles, enabling flight. No one had ever documented the presence of nociceptors, the cells that detect and transmit sensations of pain in insects. Through recent findings of nociception in larval fruit flies, challenges this and raises the possibility that some insects may be capable of feeling pain. All sensory receptors are derived from embryonic ectoderm and are integral parts of an insect's exoskeleton. They can be grouped into one of the three categories depending on function, like gustatory receptor possessing a single pore uniporous near the tip of the hair, chemoreceptors several pores, multiporous, mechanoreceptors no pore, porous. Trichoid sensillum trichoidea, with each hair formed by two cells, trichogen cell hair forming, tormogen cell surrounding socket forming several bipolar receptor cells. Basiconic sensilla basiconica, peg or cone-like process, mostly chemoreceptors. Then coleoconic sensilla coleoconica, found sunken in shallow pits, usually thermoreceptors. Sensilla ampullaceae. Basiconica, Coeloconica, ampullaceae. Ampullaceae, deep pit chemoreceptors, ambulacia, campaniformia, mechanoreceptors, non-convex hairs or bristles, shallow, round or oval pits. In longitudinal section, bell-shaped cuticular cap or dome innervated by receptor cell. Campaniform sensilla are flattened oval discs that usually serve as flux receptors in the exoskeleton. They respond whenever mechanical stress causes the exoskeleton to bend. Campaniform sensilla are found throughout the body, especially on the legs, near the base of the wings, and along sutures where two sclerites of the exoskeleton meet. Campaniform sensilla are found throughout the body, especially on the legs, near the base of the wings, and along sutures where two sclerites of the exoskeleton meet. Then placoid Sensilla placoidea, similar to campaniform, do not have hair bristles or pecs. Plate-like structure, round to oval cuticular plate surrounded by narrow membranous ring innervated by receptor cells. Chordotonal sensilla. Chordotonal organs include several types of mechanoreceptors in which one or more bipolar neurons bridge a gap between two internal surfaces of the exoskeleton. Each neuron is usually accompanied by two other cells which form a sheath, the scolopale cell and the point of attachment, the cap cell. Together, these three cells create a unit called a scolopedia that may occur singly or in groups. Common types of chordotonal organs include, commonly known as scolophore or scolopedium, consist of bipolar neuron invested by scolopale cell and an attachment cell, occurs in bundles forming chordotonal organ or scolophorous organs structured into internal integumental surfaces. Mechanoreceptors, receptors sensitive to stretching, bending, compression, torque. Stretching, bending, compression, and torque. Maintain the posture, body position with respect to gravity, detect sound waves or vibrations. Insect mechanoreceptors can be found almost anywhere on the surface of an insect body. They may act as tactile receptors, detecting movement of objects in the environment, or they may provide proprioceptive cues, sensory input about the position or orientation of the body and its appendages. These receptors are innervated by one or more sensory neurons that fire in response to stretching, bending, compression, vibration, and other mechanical disturbances. In some, mechanoreceptors produce a phasic response when stimulated, that is, they fire once when activated and again when deactivated. Other receptors generate a tonic response, firing repeatedly as long as stimulus persists. Neural processing centers in the brain or segmental ganglia interpret the combinations of tonic and phasic signals sent from nearby receptors. Tactoreceptors, touch receptors, trichoid or hair sensilla found on legs, mouthparts, and antennae. Cockroach anal cerca hair sensilla initiates escape response. Silk moth anal papillae finds oviposition site. Movement of hair triggers associated receptor cells. Dendrites of receptor cell, base of hair sensilla contain array of microtubules, tubular body and nerve impulse. Proprioceptors, maintenance of proper orientation of body parts with one another with respect to gravity, both stationary and moving insect. Provide information about different body parts to insects and tension of different muscles. Hair plate, cluster of tiny trichoid sensilla found on overlapping areas of body and head and thorax. Hair plates. Pressure differences in sideways movement. Analyzing brain, known the position of head, abdominal segments, proper posture, first and second and third, locust, vertex of head and airflow. Stretch receptors, campaniform sensillae, locusts on area where compression occur, legs, wings, halters, base of mandibles, ovipositors. Campaniform sensillae locates on area where compression occur, legs, wings, halters, base of mandibles, ovipositors. Multipolar neurons of muscles, gut, internal surface of body wall, when these tissues change in length, respond through impulse. Found on dragonfly nymph, grasshopper, and bees, wasps, moth, butterflies. Stretch receptors are multipolar neurons that usually accompany muscle or connective tissue. They are commonly embedded in integumental membranes and the muscular walls of digestive system. Some insects may stop feeding when the gut expands enough to stimulate stretch receptors in the crop. In others, oviposition may begin when mature cells stimulate stretch receptors in the reproductive system. Chorotonal organs. Bundles of Scolopedia stretched between two internal integumental surfaces found in pedicel in antennae, mouthparts, wing bases, halters, legs, abdominal segments. Also associated with the trachea, pulsatile structures, hemocoel suggests their role in intertracheal air pressure and blood pressure. Johnston's organ found within the pedicel of each antenna. In some insects, they function as proprioceptors, supplying information on position or orientation of the antennae. In mosquitoes and midges, they respond to certain frequencies of airborne sound by detecting resonant vibrations in antennal hairs. Shorter hairs near the tip of the antennae resonate to higher frequencies than longer hairs near the base. Found in pedicel of antennae of adult insects. Specialized chorotonal organ. Sound receptors, sound isoforms, potential danger, mate, prey, other members of species. Tympanic organs. Tympanal organs lie beneath a drum-like membrane, the tympanum, where they respond to sound vibrations. These ears may be located on the thorax in some hemiptera, on the abdomen in grasshoppers, cicadas and moths, or on the front tibia in crickets and katydids. Subgenual organs. Subgenual organs located in the legs of many insects. These receptors contain relatively few Scolopedia, yet they appear to be very sensitive to substrate vibrations. Insects may lack specialized sound receptors, yet they can still hear vibrations transmitted through the substrate. Chemoreceptors. Chemoreception, process by which the potential energy existing in the mutual attraction and repulsion of particles making up atoms is detected. Responsive to direct contact with atoms or molecules. Cues from environment helpful in food location and obtaining mediation of caste function, social forms, mate location, identifying noxious stimuli, threat to survival, selection of reproduction site, habitat selection. Insects have the ability to sense various chemical substances in their environment. When these chemicals are present in gaseous form at relatively low concentrations, they may be detected as odors, smells by olfactory receptors. When they are in solid or liquid form, usually at higher concentrations, they are perceived as tastes by gustatory receptors. In general, the sense of taste involves direct contact with the substrate, contact chemoreception, whereas olfaction usually implies detection of compounds in gaseous or airborne form. Remote chemoreception. Olfactory receptors, thin-walled basiconic and coeloconic pecs, identified in antennae and mouthparts of drift insects. Olfaction is mediated by chemosensilla responding to molecules or ions of a chemical in gaseous state at low concentration. Show high degree of specificity to kind of chemicals. Cuticular pores, dendrites, tormogen cell, trichogen cell, cuticle, epidermis receptor cells, nerve. Chemoreceptors, olfactory and gustatory sensilla. Sensilla were both through chemical stimuli reach nerves. Grasshopper 8 μm pecs, 20 μm diameter cavity. Smell. Olfactory receptors are usually thin-walled pecs, cones or plates with numerous pores through which airborne molecules diffuse. Dendrites of sensory neurons branch profusely within these pores and may respond to very low concentrations of detectable compounds, 6 pheromones. Some receptors respond to wide range of substances while others are highly specific. Olfactory receptors are most abundant in the antenna but may be associated with mouthparts or external genitalia. Gustatory receptors, contact receptors, basiconic and tracheoid sensilla on legs and mouthparts. Cockroaches, tips of maxillae and labial palps in buccal cavity. Mosquitoes, Cibarium, housefly pharynx. Gustatory receptors, contact receptors, basiconic and trichoid sensilla on legs and mouthparts. Cockroaches, tips of maxillary and labial palps in buccal cavity. Mosquitoes, Cibarium, housefly pharynx, honeybee, wasp tips of antennae, housefly, butterfly, bees, tibia and tarsi of 4 legs, 30 to 300 um long. Parasitic wasps, ovipositor, female judge the suitability of host. Amphid Bionodoxis indicus, Hymenoptera, distinguish parasitoid host from healthy host with ovipositor. Taste. Gustatory receptors are commonly described as thick-walled hairs, pecks or pits where the dendrites of several sensory neurons are exposed to environment through the single opening in the cuticle. Each neuron appears to respond to a different range of compounds: sugar, salt, water, protein, acid, etc. Taste receptors are most abundant in the mouthparts but may also be found on the antennae, tarsi and genitalia, especially near the tip of the female's ovipositor. Then general chemoreceptors, certain chemosensilla are responsive to high concentration of chemicals, thick-walled basiconic pecs, abundant on antennae, maxillary, labial palp, leg. Common chemical sense, high concentrations of irritant compounds like ammonia, chlorine, acids, essential oils, etc. Stimulate avoidance reactions and cleaning behavior. Insects can detect these compounds even when all known chemoreceptors have been covered or destroyed. The irritants evidently trigger a generalized response from other type of sensory neurons. Thermoreceptors. Insects, poikilothermic, no physiological control over body temperature. Temperature receptor found over body. Concentrated on antennae, maxillary, palpi, tarsi. Blood-sucking insects, mosquitoes, lice, bedbugs locate host by temperature gradient. Grasshoppers, series of paired specialized area on head, thorax and abdominal differed in cuticular structure and texture. Insects generally move away from temperature. Then hygroreceptors, chemoreceptors perceive moisture in air, found on antennae and maxillary palp. Thin-walled basiconic pecs in tribodium need low humidity and tenebrae. Locusta, cysticerca, springtails, soil-dwelling, high humidity. Then pediculus, tufts of small 4 hairs on antennae. Okay, so hygroreceptor. Then insects can all use modified forms of various sensory detectors described above to detect change in pressure, temperature, and also in some cases to detect infrared radiation, X-ray radiation, Earth's magnetic field. Hearing. Insects can hear. Insects can use sounds to communicate with other insects and to navigate their environment. Some even listen to the sounds of predators in order to avoid being eaten. Sound is produced by vibration, and insects hear by collecting and interpreting the vibrations in their environment. There are four types of insect auditory organs. Most hearing insects have a pair of tympanal organs. Think of a timpani, a large drum used in the precision section of orchestra. Like the tympanal organs consist of a membrane stretched on a frame over an air-filled cavity. When the percussionist hammers on the membrane of timpani, it vibrates and produces a sound. The insect's tympanal organ vibrates as it catches sound waves in the air. The insect also has a special receptor called the chordotonal organ, which senses this vibration of the tympanal organ and translates the sound into a nerve impulse. Insects that use tympanal organs to hear include grasshoppers, centipedes, cicadas, and some butterflies and moths. For many insects, a receptor on the antennae called the Johnston's organ collect auditory information. Sensory cells on the pedicel, which is the second segment from the base of the antennae, detect the vibration of the segments above. Mosquitoes and fruit flies hear using the Johnston's organ. The larvae of Lepidoptera and Orthoptera use small hairs called setae to gather the vibrations of sound. Caterpillars often respond to such sounds by exhibiting defensive behaviors. Some will stop moving completely, while others may contract their muscles and rear up in a flight posture. A structure in the moths or certain hawk moths enable them to hear ultrasonic sounds such as those produced by echolocating bats. The labral pilifer, a tiny hair-like organ, is believed to sense vibrations at specific frequencies. Scientists note a distinctive movement of the insect's tongue when they subject the captive hawk moths to those sounds. In flight, the hawk moths can avoid a pursuing bat by listening for their frequencies. Many but not all insects can hear sounds. Some even hear sounds that we can't hear ourselves. Insects hear through one of the four ways, the most common of which is the tympanal organs, always occur as paired organs. They are composed of a thin cuticular membrane, the tympanum, stretched across an air space of some sort and some form of connection to the nerve system. In the Orthoptera, grasshoppers and crickets, tympanum are common, those situated in different places in different species. On the first thoracic segment in grasshoppers and on the front legs in crickets. Tympanal organs also occur in the cicada, Cicadidae, Hemiptera, and some families of Lepidoptera, Noctuidae, Geometridae, and other insects. In these three forms of hearing organs are the Johnston's organ, where the movement of hairs on the antennal scape, the mosquito Aedes aegypti. Auditory hairs, these occur in some Lepidoptera larvae as well as in some Orthoptera. The pilifer, it's a unique auditory organ found only in the head of certain species of hawk moths in the subfamily Choriocambinae. Its optimum frequency is between 30 and 70 kHz, which should allow it to hear the echolocation calls of many of the larger insectivorous bats. So, Johnston's organ, tympanum, auditory hairs, the pilifer, etc. And the Orthoptera and tympanum are common. Wait a minute, once again I'm going to read this. Johnston's organ, auditory hairs, the pilifer. Then a tympanum is common in some other insects.